All right, let's start. We have spoken about microscopy for a bit. I mentioned what you need to know about microscopy as far as I remember, right? So, a couple more details about terms that you need to be aware of. So, 3 microscopy terms that you should be able to distinguish between are magnification, resolution, and contrast. So what do you need to know about magnification? It basically changes the size of the object that you're looking at. Depends on the wavelength / frequency of light, okay? And for the microscope, can be defined as magnification of the eyepieces by magnification objective, okay? So it's a product of that multiplication. Resolution. Sorry, I got one of them wrong here. This one. Sorry, resolution depends on the wavelength. I'm sorry about that. So it depends on the wavelength. It's the ability to distinguish the small details of the picture. These two are often mingled together. This is incorrect. Magnification can make the image larger, but you still may not be able to distinguish the small details. Finally, contrast is the difference in intensity. Between the specimen and the background. Okay? How dark or how bright the specimen is. So far, this is clear? Okay, let's move on. So, one thing that we do, I'm going to get rid of this. One thing that we do to increase specifically the contrast is we stain microorganisms. So, staining of microorganisms increases contrast first and foremost, but it's not the only goal. It's not the only goal. The first thing that we need to do before we stain the specimen, we need to fix the specimen to immobilize microbial cell. We can either do it using chemicals, we can do it using temperature. So what we're going to be doing either this week or next week, we're going to be fixing specimens by heating them up. Basically fry bacteria and fry them to the surface of the microscopic slide. Once they are immobilized, there are 2 approaches to staining. One approach is to use simple stains. So simple stains stain all microorganisms the same. No matter what they are, you use methylene blue, it's blue, use crystal violet, all of them are violet, use safranin, all of them are red. Simple stains can be further divided into 2 subcategories. Simple stains can be positive or negative. So positive stain stains the specimen. In our case, if we talk about bacteria, positive stains stain the cell. And background remains colorless. Does that make sense to you? Negative stains stain the background. For instance, India ink stain stains the background black while the specimen remains basically uncolored. These stains are really good for appreciating the shape, the microorganism using the light microscopy. Now, why certain stains stain the cell and others don't? Well, it turns out in positive stain there is a positively charged chromophore. Now, what is chromophore? Stain, any stain, is a chemical. So in the molecule of any stain, there is a component that's called chromophore, the component that actually gives color. Does that make sense? So that component in positive stains is charged positively. If you remember our conversation last week, we mentioned that the surface of the cell, the cell membrane, is negatively charged. So positively charged chromophore effectively binds to negatively charged membrane, staining the cell. And negatively charged chromophore of negative stains doesn't bind to the membrane, stays in the background, being repelled from the membrane. Okay, it stays in the background, stains the background essentially. Any questions on this one? Okay, if not, let's move on to differential stains. So differential stains stain different cells in the different way. Does that make sense? So 5 stains that you need to know, and I'll explain what I mean by you need to know. Gram stain, we already discussed it. It allows you to distinguish cells with a different structure. The cell wall. Acid-fast stain, used to stain mycobacteria. Those mycobacteria that are technically gram-positive but have a layer of mycolic acid on top of them. They are impervious to gram stain, so we have to use acid-fast to penetrate. Does that make sense to you? Okay, good. And then we have, I make this a pretty terrible joke all the time, that those are 3 stains that I will never ask you on the exam. And you will see why. Flagellar stain, capsular stain, and endospore stain. Quite obviously, I can't ask you, there's no point asking, what do we use endospore stain for? Well, to stain for endospores, right? So basically you have a question which already contains an answer. So nothing like that. Okay, we're good so far? Now, what I can ask you are certain details, and that's an important experimental concept about the gram staining. So when we do gram staining, usually differential stains include more than one step. Okay? So I'm going to describe, so to say, the process of gram staining and show you the roles of different chemicals. And this is what I'm going to do. We're going to have gram positives and gram negatives, and I'm going to draw kind of, bacterial cells. So there are essentially 4 steps in the gram staining. So first step when we do the gram stain is we apply, we fix microorganisms on the slide, and then we apply so-called primary stain, which is crystal violet. And this primary stain is a simple positive stain which stains both gram positives and gram negatives violet. Our second reagent is iodine, which is mordant. So what it does, essentially, it chemically modifies the stain in the cell wall, increasing the adhesion of the stain, the primary stain molecules to the cell wall. Does it change the color? No, it does not. So cell, technically, if you would look at the cells through that step, they would still be violet. But here's an interesting detail. After the mordant, crystal violet becomes pretty well stuck to the wall of the gram-positive microorganism, but not so much to the wall of gram-negative microorganism. Does that make sense? It's still there, just not as well held within. So during next step, you apply alcohol, which is Decolorizer. So Decolorizer, alcohol, is supposed to remove the stain, okay? And it does so from gram-negatives, but not from gram-positives. Does that make sense? Because crystal violet is pretty well stuck to the cell wall, okay, so bless you, so it stays there, but gram-negatives lose the color. And finally I have the last step, which is the application of counter stain or secondary stain. Safranin. So safranin stains gram-negatives red, and gram-positives, due to the really intense color, bless you, remains violet. So at the end of the day, you can have a sample, even a mixed sample of gram-positive and gram-negative microorganisms, and you can clearly see that your sample contains them both. Does that make sense to you? Good? So this is the steps of gram stain. What I can ask you about the roles for different stains. Like, what is the role of alcohol in the whole process, or what is the role of iodine? Just pick the answer. Does that make sense? Okay, good. Now,